Okay, so now that we have sort of introduced the idea of long-term potentiation, where long-term potentiation, it really just refers to the strengthening of a synapse between two neurons, and that is really the biological basis of all learning. When you are learning something, whether it's some new fact or you're learning a person's name that you've never met before, you are literally either creating new synapses between neurons or you are strengthening synapses that already exist. And so that process of strengthening synaptic connections is what we call long-term potentiation. And so we're going to focus a little bit more on that here in the next couple of slides. So let's take a look at this process of long-term potentiation and sort of get an idea for what it looks like in a cell. So the picture here that is shown is from an experiment in the 1980s, one of the first to demonstrate this principle of long-term potentiation. And so let's just sort of identify first, before we walk through this, all the parts that we're going to be talking about. This is actually this cell here. This is the same cell just being shown twice, so let's just sort of draw a line down here between the two here. We're going to talk about the one on the left first, and then take a look at the one on the right afterwards. So this is actually a particular type of neuron. It's called a pyramid cell, which is found in parts of the hippocampus. And that's important because what have we said before about the hippocampus? The hippocampus is a crucial brain area for learning and remembering, and it's particularly in the hippocampus that you see long-term potentiation, which is really what we would expect, right? If long-term potentiation, the strengthening of synaptic connections between neurons, is the biological basis for learning and memory, then it makes sense that you see this process in a part of the brain that we now know to be associated with memory, and that's the hippocampus. So this is looking at a pyramid cell, and you can sort of see that it is called that because of the shape, the particular shape of the cell body here, right? It sort of looks like a triangle or like a pyramid. And so it's the cell, you can see it's got its axon here coming away from the cell, it's away from the cell body, some dendrites coming off of it, and then one sort of really big massive dendrite coming up here. This is actually a dendrite. I'm just going to draw my line going up here. And well, I don't want to draw an arrow going that way. These things here are dendritic spines, they're called. These are just sort of enlarged little parts that stick out from the dendrites, and basically are parts of the dendrite that synapse with the axons of other neurons, right? So here, for example, is sort of the synapse that we'll be focusing on. The synapse between a presynaptic neuron, that would be this guy here, the blue one, would be the presynaptic, and then this pyramid cell would be the postsynaptic neuron. So this presynaptic neuron, the axon of this cell, is forming a synapse with the dendritic spine of this pyramid cell, this postsynaptic neuron. And so what this picture is showing here is the result of an experiment from the 1980s by Kelso and colleagues. And they found that if they used a microelectrode, that's what this is here, this is sort of supposed to show a microelectrode that you can actually insert into a cell. It's a very little tiny electrode that you can sort of stick into a cell and run a very small electrical current through it. Now what does that do? Running a small electrical current into a neuron will cause depolarization. It will cause this cell to trigger an action potential if you run enough electricity through it, right? So if you stimulate this cell enough, it will generate an action potential. And what these guys found is that if they used this microelectrode to sort of artificially depolarize this neuron, right? So they're the ones depolarizing it. They're the ones that are stimulating this neuron, as opposed to what would be sort of natural depolarization, right? If this cell were to be stimulated by just another neuron in the brain somewhere. So if they depolarize this neuron, And then they stimulated an axon that formed a synapse with this neuron. So they stimulate this cell as well, this presynaptic neuron that I pointed out before. If they did both of these things at around the same time, what they found was that the synapse between this presynaptic cell, this blue neuron, and the synapse between this and this dendritic spine, this particular synapse, became stronger. It strengthened. Which means what? Given what we said before, it means that following strengthening, if this neuron, this blue neuron here, were to be stimulated and release neurotransmitters into the synapse that were to be picked up by this cell, because the synapse has been strengthened, this pyramid cell, this postsynaptic cell, will be even more likely to generate an action potential in response to this cell. So after strengthening, this pyramid cell will be more likely to generate an action potential when it's been stimulated by this cell, compared to over here, before this synapse had been strengthened. You know, if this blue neuron had been stimulated and it released neurotransmitters, maybe sometimes this pyramid cell would generate an action potential in response to that, but not all the time. This would have been a fairly weak synapse. But after this strengthening process, these two neurons, have essentially kind of been glued together. This synapse has been strengthened a whole lot, so that now when the blue neuron has been stimulated, that almost always causes this pyramid cell to also be stimulated and to generate an action potential in response to that. And so this is what we've been saying is long-term potentiation, is the strengthening of a synapse between two neurons. And so an easy way to sort of think of long-term potentiation is in terms of this popular saying that neuroscientists have, and that is that neurons that fire together wire together. So what does that mean in the context of this particular picture? Notice what the researchers had to do to cause long-term potentiation in order to cause this synapse to become stronger. It didn't just require them to stimulate this blue neuron. Two things had to happen. One was they had to stimulate this blue neuron. Do I have an eraser? I do have an eraser. Nice. So let me just get rid of this. One of the things they had to do was to stimulate this blue neuron. But that wasn't enough. That in and of itself would not cause long-term potentiation. What was the other thing they had to do? They also had to stimulate this pyramid cell. They had to stimulate both the presynaptic cell as well as the postsynaptic cell. And so that's where we get this saying. Neurons that fire together wire together. The blue neuron and the pyramid neuron fired together. And so now, through the process of long-term potentiation, are now wired together. Wired together in the sense that their synapse has been strengthened. Okay, so now that we have seen sort of what long-term potentiation looks like, and a little bit we've introduced what is required for long-term potentiation to take place, we're going to start getting even more in depth with this in terms of talking about what exactly is happening at a molecular level that causes this process of long-term potentiation, that causes this strengthening of synaptic connections. So we've said already that really two things have to happen in order to see long-term potentiation, in order to see this strengthening between the strengthening of the synapse between two neurons. What are those two events? Those two events are, one, there has to be activation of the synapse, which basically means stimulation of the presynaptic neuron, right? That was in the previous picture. Let's go back here. That's this, right? Right here. This is activation of the synapse by stimulating the presynaptic neuron. So that's one thing that's necessary to happen. The other thing that's necessary is depolarization of the postsynaptic neuron. And so that is, if we just again go back to this picture, that's this, right? So the two things that have to happen in order for long-term potentiation is activation of the presynaptic neuron and activation of the postsynaptic neuron. And again, back to this idea that neurons that fire together wire together. Now the reason that these two things are important, or the reason for long-term potentiation, has to do with one of two types of receptors that we're going to be talking about here. As I mentioned before, there are really two types of receptors that are important for long-term memory. One of those is something called an NMDA receptor. And if you remember, what are receptors? Receptors are binding sites in the membrane of a postsynaptic neuron, usually in the dendrite, right? So this would be something that's located in the dendritic spine of the postsynaptic cell. So there's little receptor sites or binding sites that are located in the membrane of this postsynaptic cell that are going to pick up neurotransmitters that get released by the presynaptic cell. Okay, so the first receptor that we're going to talk about is what's called an NMDA receptor. And the first neurotransmitter that we're going to talk about, well, actually this might be the only neurotransmitter that we talk about, one that's really important for long-term memory, is something called glutamate, which I mentioned before. You can think of it as glue. And I think my time is almost up on this video, so I'm going to stop this video, and I'll be back with the next one in just a second.